Bismillahirrahmanirrahim. Today I am going to discuss cardiac muscle cells. There are two major types of cardiac muscle cell. Myocardial contractile cells. These are the myocardial contractile cells, and the second one is myocardial conducting cell. So these are the myocardial conducting cell. We will discuss them in the next slide. Autorhythmicity. Atomicity, or chronotropism. In the myocardium, autorhythmicity is the ability of the cardiac muscle to depolarize spontaneously without external electrical stimulation from the nervous system. Regardless of the pathway, cell in the SA node spontaneously depolarize, resulting in contraction approximately 100 times per minute. This native rate is constantly modified by the activity of sympathetic and parasympathetic nerve fibers, so that the average resting cardiac rate is about, in adult human, is about 70 beats per minute. The ability of the cardiac muscle to initiate an electrical potential at a fixed rate that spread rapidly from cell to trigger the contractile mechanism. This property is known as autorhythmicity. So this is the definition that the cardiac cell are automatically initiate the action potential. The myocardial contractile cells. The myocardial contractile cells constitute the bulk, 99% of the cell in the atria and ventricle. So the atria and ventricles are composed of 99% of the cardiac muscle or contractile cells. Contractile cells conduct impulse and are responsible for contraction of cardiac muscle that pump blood through the body. The heart receives blood from the body and from the lung, and then by contraction and relaxation, the heart pumps this blood into the general circulation throughout the body. So this is the main function of heart. The atria and the ventricular type of muscle contract in much the same way as the skeletal muscle, except that the duration of contraction is much longer. The duration of contraction of cardiac muscle is longer than the duration of contraction of the skeletal muscle. Otherwise, both contract in the same way. The myocardial conducting cell. 1% of the cell form the conduction system of the heart. So the conductive system of the heart is composed only of 1% of the cell. They are generally much smaller than the contractile cell and have few of the myofibril or filaments needed for contraction. Their function is similar in many respects to neurons, although they are specialized muscle cell. In function, they are similar to neuron, but they are not neuron in nature. They are actually modified cardiac muscle cells. The conduction in this system occur in the pathway. First, from SA node to the AV node. This is the SA node, and from SA node through the internodal pathway, the current is passed from SA node to AV node. and from AV node to the bundle of Hess. From bundle of Hess, the current is passed then to bundle branches and then to the Purkinje fibers. The Purkinje fibers deeply innervate the cardiac muscle cell, and when current is spread along this pathway, it causes contraction of the muscle. The internodal pathway are three in number: the anterior, medial, and posterior. Structure of cardiac muscle. Compared to skeletal muscle, cardiac muscle cell are cardiomyocytes are considerably shorter with much smaller diameter. They are short than the skeletal muscle and they have smaller diameter than the skeletal muscle. Cardiac muscle also demonstrate striation. You can see that there are striation in the cardiac muscle. The alternating pattern of dark A band and light I band attributed to precise arrangement of the myofilament and fibrils that are organized in sarcomere along the length of the cell. What is sarcomere? We know that sarcomere is space between the two Z lines. So here you can see that this is the Z line and we have myofibril between these two Z lines, and they have the same characteristic or structure or arrangement like in skeletal muscle. These contractile elements are virtually identical to the skeletal muscle. Transverse tubules penetrate from the surface plasma membrane, the sarcolemma, to the interior of the cell, allowing the electrical impulse to reach the interior. The T tubule, you can see that they are originate from the sarcolemma or membrane of the muscle cell and penetrate deeply into the cardiac muscle cell. In this way, they are connected with the exterior of the or extracellular fluid. The T tubule are only found at the Z disc, where in skeletal muscle they are found at the junction of the A and I band. This is the Z line, you can see, and the T tubules in cardiac muscle are present at a level of Z lines, while in skeletal muscle these tubules are present in I band and A band. So in this way you can see that the skeletal muscle have two T tubules in one muscle cell. On the other hand, the cardiac cell have only one T tubule. Here you can see that this is the T tubule which originate from the sarcomere and it is found at the site where the Z line is located. Also, the two cells join each other by intercalated discs, and these cells contain much mitochondria and less T tubules. The T tubule number is less than the skeletal muscles, and it contains much of the mitochondria. The cells are also joined together by the gap junction or intercalated discs. Structure of cardiac muscle. Therefore, there are one half as many T tubules in cardiac muscle as in skeletal muscle. The number of T tubules are less than the skeletal muscle by half numbers, because in cardiac muscle T tubules are present at Z lines, and in skeletal muscle the T tubules are present at the level of I and A bands. The sarcoplasmic reticulum store few calcium ions. So most of the calcium ion must come from outside the cell. The result is a slower onset of contraction. The contraction of the cardiac muscle is slow regarding or comparing with the skeletal muscle because it contains less amount of calcium in the sarcoplasmic reticulum, and during contraction most of the calcium is drained from the extracellular fluid. It also contains plentiful mitochondria because the heart remains active throughout the life and need more energy. Structure of cardiac muscle. Typically, cardiomyocyte, the cardiac muscles are also called cardiomyocyte, have a single central nucleus. You can see that there is a single central nucleus. Cardiac muscle cell branch freely. They are branching. Here you can see that one cell, when they combine with each other and then they branch, they make a branching fiber and they join each other. A junction between two adjacent cells is marked by a critical structure called an intercalated disc, which helps support the synchronized contraction of the muscle. Here you can see that this is the cardiac muscle cell and this is another cardiac. When these two cells join each other, at the joining point there is a structure which is called intercalated disc. It is present at the junction of the two cells. This structure is also called gap junction and it makes communication between the two cells. Lattice work. Cardiac muscle fiber arranged in a lattice work with the fiber dividing, recombining and then spreading again. Here you can see that these fibers are dividing, making branches. They join with each other and then divide and then branch. So this pattern of arrangement is called lattice work. This is just like the hairstyle of a girl's desmosomes. So desmosomes are the sarcolemma from adjacent cell bind together at the intercalated disc. The two cardiac cells join with each other at the site of intercalated disc. They consist of desmosomes. At the intercalated disc we have cells which are called desmosomes, which are specialized linking protoglycan tight junction and large number of gap junctions that allow the passage of ion between the cell and help to synchronize the contraction. At the level of intercalated disc, these proteins are present. This is cell 1 and this is cell. So these two cells are joined together by the desmosome. They are protein in color. They are the green color protein are actually located intercellular and they are called cadherin. The red one is desmosome and the blue one is the keratin, and they actually connect the two myocardial cells tightly with each other. That during contraction they are protected from derangement. During contraction also at this point the gap junctions are present and they have tight connection with each other and also sharing the information with each other. That at this level the ions are passed through the gap junction from one cardiac cell to the other cardiac cell. These proteins are proteoglycan in nature and they actually make the cell, they keep the cell tightly together. Intracellular connective tissue also help to bind the cell together. These are the intracellular tissue. Gap junction. At each intercalated disc, the cell membrane fuse with one another in such a way that they form permeable communication junction, which is called gap junction, that allow almost totally pre-diffusion of ion. You can see that these are the gap junctions. They have no gates and they allow ions from one cell to the other cell freely. Ions move with ease in the intracellular fluid along the longitudinal axis of the cardiac muscle fiber, so that action potential travel easily from one cardiac muscle cell to the next past the intercalated disc. So these gap junctions actually allow the flow of ion from one cell to the other cell along the longitudinal axis of the cardiac muscle cells, and in this way they share one cell share its information with the other. Cardiac muscle is sensatium. The heart actually is composed of two sensatium, the atrial sensatium that constitute the wall of the two atria, the right atria and the left atria, and the ventricular sensatium that constitute the wall of two ventricles, the right ventricle and the left ventricle. So sensatium is actually sensatium means that when a single cell is activated, it contract the whole belly. The atria separated from the ventricle by fibrous tissue that surround the atrioventricular valve opening between the atria and ventricle. This the atria interiorly is covered by connective tissue. This connective tissue actually plays the role of insulation. That the current when the atria is stimulated or when contraction in the atria occur, this action potential or stimulation doesn't pass directly from the atria to the ventricles. This because these connective tissue insulate the gap junction, and the conduction from atria to the ventricle is only through the AV node. The AV, there is a delay in the AV node, a brief delay of 0.1 second, and this allows the atria to contract before the ventricle. Normally, potential are not conducting from the atrial sensatium into the ventricle sensatium directly through this fibrous tissue. They insulate the gap junction. Instead, they are conducting only by way of specialized conductive system called the AV bundle. This division of the muscle of the heart into two function sensatium allow the atria to contract a short time ahead a ventricle contraction, which is important for effectiveness of heart pumping. The atria contract before the ventricle. When the atria contract, the ventricle at that time are relaxing. So this allows the flow of blood from the atria to the ventricle, and this also allows the ventricle to fill with blood and form that blood into the circulation. So this delay in the atria and ventricular contraction is necessary for the proper pumping of blood throughout the body. If both will contract at the same time, so there will be no pumping of blood into the general circulation and pulmonary circulation. So this delay is important, and this delay actually occur due to the delay or in the sinoatrial node, atrioventricular node. So then the current pass through the bundle of to the bundle branches, and then the ventricle contract and pump the blood into the general circulation. Thank you. Please subscribe the channel and press the bell icon for in-time notification, and leave your value